Hi everybody, so today we will be talking about PCR or the polymerase chain reaction. So the polymerase chain reaction is used to amplify large quantities of DNA from a small quantity. It can be also used to analyze single DNA fragments from a mixture of DNA, and it can also be used to alter DNA sequence through site-directed mutagenesis. So the components of a PCR are definitely the template DNA, or the DNA which acts as your template. You'll need primers to specifically amplify a particular portion of that template DNA. You will need a polymerase to basically replicate the DNA. You will need nucleotide triphosphates, which are required for DNA replication. You'll need a buffer which contains magnesium, and of course you will need a thermocycler. So this is a certain brand of thermocycler, which will basically cycle temperatures up and down to conduct all the steps in a PCR reaction. So this is an overview of the PCR temperature cycling reaction. So the first thing that happens is once you've put all the components together in your little microfuge tube, you then the PCR basically heats up to 94 degrees, and during this the DNA denatures, which means the strands get separated. Then the temperature is lowered to around 55 degrees to allow the primers to anneal to the DNA. Then in the third step, extension occurs, by which the primers are extended to form a new strand of DNA. So I'm going to show you the movie right now. We can see the DNA denaturing, and now you see the primers annealing, and then the Taq polymerase creating the new strand, and then it happens over again. So denatured, then primers anneal, and then you have the extension phase in which the new strand is created. So over the course of around 30 to 35 cycles, what happens is that the DNA becomes replicated. So from 4 copies, you get 8 copies, you get 16 copies, and then finally 32 copies until you can get 1 billion copies in theory after around 30 cycles. So the beauty of this process is it's basically very simple. It takes around 2 hours to run a PCR reaction, and it amplifies specific parts of DNA as defined by your primers, and that's very important. So after you've run the PCR reaction, you take your little microfuge tube off of the thermocycler, and then you run the DNA in a gel. So ideally you want to see fragments like this, which are single fragments that have been amplified. So there are some variables in a PCR reaction. For instance, you can vary the cycle times and the temperatures. You can vary the primer design. You can add components to your buffer. And of course, there are different types of polymerases that you can use. So what happens is that when you vary the cycle number, there's a plateau effect. So PCR basically works really well up to 30 cycles, because after that the polymerase doesn't work efficiently anymore. Also, the nucleotide triphosphates also get depleted. Sometimes if you vary the cycle times and temperatures, you get better results. Specifically, during the extension phase, you can vary the number of cycles and the temperature. Primers. So when you design primers, you have to keep in mind that the optimal length of primers is around 18 to 28 bases. And usually they have a temperature melting range, or a TM, of 52 to 58 degrees centigrade, and a GC content of around 50 to 60%. Buffers. So you usually add magnesium to the buffer because magnesium is a cofactor for the polymerase and helps it to work more efficiently. Sometimes you add substances like DMSO, which is a helix destabilizer, and it helps to break up DNA with high GC content because guanines and cytosines are held together by 3 hydrogen bonds. So though you have to break 3 bonds as opposed to breaking the 2 bonds of A and T. There are different types of polymerases that you can use, and they can be either native polymerases that can be found in nature like TAC, or they can be cloned or basically souped up polymerases. So if you look at, let's say, the error rate of TAC polymerase, there's 1 error every 10,000 nucleotides, whereas for PFU polymerase, you only get 1 error per million nucleotides. So it does matter which polymerase you use in your reaction. So a typical PCR reaction is made in a 50 microliter volume, and we've talked about the components before. You can also half the volume or double the volume as long as you keep the ratios of the components constant. And as I said before, the beauty of this reaction is that within a space of around 2 hours or so, you can amplify specific targets on your DNA.